In this video, we're going to look at cell membranes and how they're constructed in order to allow movement of materials into and out of cells. Just to refresh your memory, let's recap about how a membrane is constructed. Remember that membranes are made up of phospholipids, and phospholipids have two fatty acid chains hanging below a glycerol molecule and one phosphate group above the glycerol molecule. Remember that the fatty acid chains, because they don't have polar bonds, are very hydrophobic. The phosphate group, on the other hand, is very hydrophilic, making the molecule as a whole amphipathic, or having both hydrophilic and hydrophobic regions. When these molecules are arranged in a membrane, the tails point to each other and form a hydrophobic region within the membrane, and the hydrophilic region facing inside or outside of the cell. The way that scientists now understand how a membrane is constructed is based on what is known as the fluid mosaic model. In this model, some of the molecules move around laterally in relation to each other, and they move quite a lot. Among the phospholipids are proteins that are embedded into the cell membrane. If the protein is just on the membrane's surface, we call that a peripheral membrane protein. If the protein goes all the way through the membrane, we call it an integral membrane protein. Let's take a moment to look at why some proteins sit on the surface and some integrate through the membrane. Remember that proteins are made up of amino acids, and R side groups of an amino acid is either hydrophilic or hydrophobic. As the protein folds up into secondary, tertiary, and quaternary structures, hydrophilic regions and hydrophobic regions emerge. The hydrophilic regions will tend to locate themselves on the surface of the membrane, where they're in contact with either the liquid on the outside of the cell or the cytosol on the inside of the cell. In the case of transmembrane integral proteins, they have regions that are hydrophilic on both sides of the cell membrane and a hydrophobic region in the middle. According to the fluid mosaic model, phospholipids and proteins may move around laterally on the surface of the membrane. However, these molecules rarely flip over. You can see why this must be the case. In order to flip over, an integral membrane protein would need its hydrophilic region to pass through the extremely hydrophobic region within the membrane. It doesn't do this because the hydrophilic region is so much more strongly attracted to the water on the surface of the cell than it is to the hydrophobic regions inside the membrane. Some nice experimental evidence for the fluid mosaic model came from a study by Fry and Edidin. In their study, they marked proteins differently on mouse cells and human cells. They then made a hybrid cell by fusing together the mouse and human cells. Shortly after creating this hybrid cell, they were able to observe that the proteins on the surface of the cell had become evenly distributed around that final cell. If the proteins were unable to move, as they do in the fluid mosaic model, different regions of the final fused cell would have had either mouse or human proteins. However, mouse and human proteins were evenly distributed over the entire cell surface, lending support to the fluid mosaic model. The ease or rapidity with which phospholipids and proteins move across the surface of a cell is called the membrane fluidity. If the membrane is highly fluid, then the movement is much more rapid, but the membrane risks breaking apart. If the membrane is too solid, it can't function properly and movement of the molecules is hindered. There are a number of factors that affect membrane fluidity. The first is temperature. When a membrane is warm, there's more thermal energy of the molecules. They're less stable and they tend to move more, resulting in higher fluidity at those temperatures. When the temperature is cold, the membrane solidifies somewhat, in the same way that bacon fat solidifies when you cool down the frying pan. The second factor is the presence of unsaturated versus saturated fatty acids as components of the phospholipids. When a membrane has primarily saturated fatty acids, the fatty acid tails form straight bonds and are packed very close together. This close packing results in more intermolecular interactions, such as van der Waals forces, that stabilize the membrane, resulting in a more viscous or solid membrane. When the phospholipids have unsaturated fatty acids, the double bond introduces a kink to the tail. As a result, the phospholipids don't sit so closely adjacent to one another, and intermolecular interactions are reduced, increasing the membrane fluidity. The third factor is the presence of cholesterol within the membrane. Remember that cholesterol has a hydrophilic end and a large hydrophobic end, and therefore packs adjacent to the phospholipids. In a membrane, cholesterol plays a dual role. When the membrane is warm, the cholesterol reduces its fluidity. When the membrane is cold, however, cholesterol increases or maintains a high degree of fluidity. You can think of this like the antifreeze you put in a car. Antifreeze prevents the coolant from freezing on a cold day, but also prevents it from boiling when hot. Similarly, cholesterol maintains appropriate fluidity under both warm and cold conditions. The last factor affecting membrane fluidity is the length of the fatty acid chains. Phospholipids with short tails have less intermolecular interactions and are therefore less stable and more fluid. Phospholipids with longer fatty acid chains have more van der Waals forces that stabilize the membrane and are therefore less fluid. Let's just take a moment to appreciate the variety of proteins that exist within the membrane. Each protein has a specific function, and these functions are highly varied. Some proteins act to transport materials into or out of the cell. Some proteins on the cell surface are enzymes and speed chemical reactions happening on the surface of the membrane. We'll see some examples of this when we look at how energy is processed by mitochondria. Other proteins transmit signals from one side of the membrane to the other. They receive a signaling molecule on one side of the membrane and pass the signal to the other side. Some proteins are responsible for cell-cell recognition. Most of our cells have glycoproteins on the surface, and the oligosaccharide chain is unique to our cells. Our immune system identifies those cells as being legitimately part of our body. If our immune system identifies a cell that does not have the correct oligosaccharide on the surface, the immune system will kill that cell. As we've seen in a previous presentation, some proteins on the cell surface are responsible for connecting one cell to another. Additionally, proteins are responsible for attaching and stabilizing the cell membrane to both the cytoskeleton inside the cell and the extracellular matrix outside the cell. Okay, let's take a look at some of the factors that affect membrane permeability. We're going to look at both characteristics of the solutes that are moving through the membrane as well as characteristics of the membrane itself. If you haven't yet watched the presentation on osmosis and diffusion, I encourage you to do so now. In order to move across the membrane, molecules must be able to fit between the molecules of the membrane itself. Therefore, small molecules are much more likely to cross the membrane than large molecules. As molecules pass through the membrane, they must pass through the hydrophobic region. In order to do that, they need to be hydrophobic themselves. Hydrophilic molecules will tend not to cross through the membrane because they'll be attracted to the water on either side of the membrane. Because the water can't enter the membrane, the hydrophilic solutes will also tend not to enter. Therefore, we expect that as the charge or polarity on the solute increases, the chance that it will pass through the membrane decreases. We therefore expect to see nonpolar molecules moving through the membrane more quickly than charged molecules, or especially much faster than ions. So just to summarize, small molecules move through the membrane much more easily than large molecules, And polarity or charge hinders the movement through the membrane, and therefore the stronger the charge, the less movement we have. Okay, so we've looked at some of the characteristics of the solute that allows or prevents it from traveling through the membrane. Let's look at some of the characteristics of the membrane itself. Membranes that have unsaturated fatty acid tails pack more loosely together and therefore have more space between them for solute molecules to travel. If the fatty acid tails are saturated, there is less space between them and solutes have a harder time traveling through the membrane. If the fatty acid tails are short, there's less distance for the hydrophilic molecule to travel between the hydrophilic regions on both sides of the membrane. It's therefore more likely to do so through a narrow membrane than it is through a wide membrane with longer fatty acid chains. Finally, high temperatures result in higher fluidity, and this additional movement of membrane molecules allows higher permeability. Up until now, the process of getting solute molecules across the membrane has relied on diffusion alone. When there's a strong diffusion gradient, that is, a large difference in concentration between inside and outside the cell, solutes will tend to diffuse more quickly. However, you may have noticed that some molecules, particularly large or charged molecules, only rarely move through the membrane. In order to transport these through the membrane, the cell must use other mechanisms than just diffusion directly through the membrane. This is where one of the roles of integral membrane proteins come into play. Channel proteins are membrane proteins that simply serve as like a hole through the membrane. Solutes can pass freely through the channel protein, but they always go in the direction dictated by diffusion. That is, they always go down the concentration gradients from a region of high concentration to a region of low concentration. If you look at this upper picture, you see that you see molecules moving through a channel protein. In reality, the channel proteins are very specific to particular solutes and only allow that particular solute to pass through. In contrast to the static channel proteins, carrier proteins are much more dynamic. In order to transfer a solute molecule from one side of the membrane to another, carrier proteins must bind or somehow interact with the solute molecule in order for them to pass through the membrane. When a carrier protein binds a solute molecule, it changes its shape in order to allow it to be released on the other side of the membrane. Now, both of these mechanisms are considered to be passive transport. In passive transport, the proteins simply serve as a conduit for the solute. However, the solute is always traveling in the direction of its concentration gradient. Because it's traveling down the concentration gradient, no energy is required. Some passive transport occurs through gated channels. This type of transport is still passive because the movement of solutes still goes down its concentration gradient. However, the channels are gated, that is, some stimulus is required for them to open. In order to transmit a nerve impulse, a particular charge across the membrane is responsible for opening the ion channel. In other cases, neurotransmitters are the stimulus required to open those channels. Often, cells can't rely on diffusion or passive transport alone, because with passive transport, solutes are moving from areas of high concentration to low concentration. If a cell needs to add solutes to a region where it's already highly concentrated, energy is required. Most cells maintain an electrical charge across their membrane, with inside the cell being slightly more negative than outside the cell. The sodium-potassium pump shown here helps maintain this electrical gradient and is a really good example of active transport. This pump binds 3 sodium ions from the inside of the cell and uses energy in the form of ATP to release them outside the cell. In order to complete its cycle, it then moves 2 potassium ions from outside the cell and releases them inside. Because of this 3-for-2 exchange, outside the cell becomes progressively more positively charged than the inside, and the action of this pump maintains this difference. Up until now, we've only looked at chemical gradients. That is, which region has a chemical more highly concentrated and which region has it at lower concentrations. However, sometimes we need to also consider electrical gradients. Which side of the membrane has a relative positive charge and which side is negatively charged? This is important because just as molecules move down their concentration gradient, they also move away from regions with the same charge as they have. Thus, this combination of forces is called the electrochemical gradient and plays an important role in storing potential energy across a membrane, as we'll see later. So in animal cells, the sodium-potassium channel can maintain an electrochemical gradient across the cell membrane. In plants and other organisms, proton pumps are very common and serve a similar purpose. Cells can use a strong electrochemical gradient as a sort of battery. The difference in charge across the cell membrane can be used to drive other processes. Cotransport is a good example of this. In the example of cotransport shown here, energy in the form of ATP is used by a proton pump to pump protons from inside the cell out. As a result, there's a strong electrochemical gradient with the protons trying to get back into the cell. A cotransport protein takes advantage of this gradient and uses the affinity of the protons to go into the cell in order to drive the transport of sucrose into the cell against its concentration gradient. Notice that the sucrose is being concentrated by the cell and is therefore going against its concentration gradient. This usually requires energy. But instead of using ATP directly in the sucrose cotransporter, the ATP is used by a separate proton pump in order to drive the protons out. The electrochemical gradient itself provides the energy for the proton sucrose cotransporter to operate. So, just to very briefly review the different types of transport of small amounts of material, with passive transport, the material always goes from a region of high concentration to a region of low concentration. That is, it goes down its concentration gradient. The simplest of these is diffusion, but diffusion is generally slow across the plasma membrane except for small uncharged molecules. Larger and/or charged molecules generally require facilitated diffusion, and this can either be through channel or carrier proteins. If material needs to be transported against its concentration gradient, that is, from a region of low concentration to high concentration, active transport is required. Active transport always requires the input of energy, and sometimes this energy is used directly, and other times it's used indirectly, as in the case of cotransport. Often, the cell needs to transport materials out of the cell in bulk. It does this through a process called exocytosis. During exocytosis, transport vesicles move the proteins or other materials to be exported to the cell membrane. When the lipid bilayers touch one another, they start to fuse, eventually dumping the contents of the vesicle outside the cell. There are several ways in which materials are brought into a cell in bulk. In phagocytosis, very large materials can be brought in. Pseudopodia, or false feet, extend from the cell surface around the food item and eventually engulf it. This happens through extensions of the actin filaments of the cytoskeleton that we looked at in a previous presentation. After the food item is ingested, it's sequestered into a food vacuole until it can fuse with a lysosome, which contains digestive enzymes to break the food down. In pinocytosis, small pits form in the surface of the cell, and whatever solutes are in that region are brought into the cell. Cells use receptor-mediated endocytosis to bring in specific molecules to the cell. Receptors for the target molecule are concentrated in regions of the membrane, and as these are brought into the cell, the contents of the vesicle is mostly the target molecule. Both pinocytosis and receptor-mediated endocytosis make use of coated pits. The coat protein allows the cell membrane to pinch inward and form a vesicle. When the contents of the vesicle have been digested by the cell, the coat proteins are recycled back to the cell membrane.